It's my pleasure to introduce Pedro Martinez, who's the director of the Center for Marine Biodiversity at the Senckenberg Institute in Wilhelmshaven. And Pedro is going to take us on to the wonderful worlds of Harpacticoid Copepods.
Only. So what is that? At the bottom. Thank you, Anne. So this is about the taxonomy and biogeography of Meiofauna Harpacticoid Copepods, but I will talk about copepods in general. First of all, to mention that there are two important homepages if you want to read more about copepods. One is the marinespecies.org/copepoda, where you will find this homepage, and there is a lot of resources there. So, for instance, there are links to books with and literature which is not copyrighted anymore, and you can download it from there. There's another homepage which is monoculus.org, which is the homepage of the World Association of Copepodologists. So something like that also exists. The first copepod that were described, also they are known also from Aristotelic times, was in 1554. It is a parasite on the bluefin tuna, that was collected and was identified and depicted here, something similar like that. And it took about 100 more years for the second copepod to be formally described. This is in 1684 by Reddy. This was a parasite on an ascidian. And in 1671, this is a parasite also on a fish. As you can see, parasitic copepods were described first. The first free-living copepods were described from freshwaters. This is from Blanca, the first cyclopoid copepod, and also from the plankton, from the marine plankton. These are these drawings from Gunerus, 1770, which describe this Monoculus finmargicus, which is actually now called Calanus finmargicus. It's one of the most important calanoid copepods in the plankton in the North Sea. We have also from Gisbrave, but also from Haeckel and other workers, fantastic drawings on copepods. They are indeed colorful, if you look at them alive, but they lose the pigments if you fix them with formalin or with ethanol. And so you see that the history of copepods is and was during 700 years related to the question of how the parasitic copepods are related to the free-living copepods. And this was very relevant for the classification of the copepods if you look at a historical perspective. So the first classification was done according to the mouth parts. So there were some of them, the gnatostoma with large oral cavity and fitting mandibles with palps. The siphonostoma, they have like an oral cone, there is a parasite, and stylet-like mandibles, or the poecilostome, which has no mandibles at all. But other workers like Gisbrecht, they found that the body tachmosis, so how the body is organized, this is the head and the thorax and the abdomen. You can see this type of copepods, they have this leg incorporated into the thoracic segment, but this other type of copepods has this leg incorporated into the abdomen. So this kind of separation of the body was most important for the high-level classification of copepods for other workers. To make it short, because there has been a couple of many proposed classification of copepods, this is the system that we are more or less using today from Hoyes and Boxall, 1991. They published a monograph of copepods, and this is a system comprising 10 orders. Important for us are these orders. The calanoid copepods, these are the most important marine planktonic copepods. The Harpacticoid copepods, these are the most important benthic copepods, interstitial, living in the sediment. We have the siphonostomatoid copepods. These are all of them associated with invertebrates or fishes. These are the most important copepods in hydrothermal vents, of course, because they are associated with the megafauna there. And then we have also in the abyss also some other orders, rare species, not very represented by many organisms like the Meiofauna and the Cyclopods, that they are living in what we call the hyperbenthic community. So they are not really in the plankton above the bottom, they are also not in the sediments, but they are living in the water column above the sediment, so 1 meter, 2 meters, 3 meters above the sediment. There you can find them. This is how they look like. These are different pictures of copepods. Here you can see that there's something hanging here, so what's that? These are eggs. So these 3 animals are females. Females carry the eggs in an egg sac. You can see that some of them, they have a lot of eggs, so this is an R strategy. So they have a lot of eggs, a lot of outcomes. This is a deep sea copepod carrying only a couple of eggs. This is a hydrothermal vent copepod carrying only a single very big egg. And this is telling you already what is the strategy in reproducing of these copepods. So this individual, this specimen, is investing a lot of energy to 1 single or 2 single eggs in order to pass this energy to the larva. I wanted to show you also an example of a very nice deep sea copepod, armed with a lot of spikes here. We have no idea what they're doing with these spikes. This is Ceratonotus steiningeri. You will hear about it also in other slides. This is from the CCZ actually. So in summary, copepods, there are about 14,000 species described. Some 10,000 species are marine. And there are 10 orders. Harpacticoida is benthic, mainly benthic. Cyclopods are mainly freshwater, but also some marine. Siphonostomatoids are associated with fishes and invertebrates, and calanoids are planktonic. They occur from the deep sea, 11,000 meters, to the top of the Himalayas. Some of them, they're living in the ice because they are feeding on algae, living in the ice. Different ranges of temperature from -2 degrees to hot springs, hydrothermal vent, cold seeps, groundwaters, they are actually everywhere. Here you see me some years ago with my daughters collecting some copepods here in the riverbed in Brazil. So they are in the phreatic waters, in the groundwaters. You can find them also in bromeliads. You can find them in the marine ice. You can find them in anchialine caves. You can find it in chemosynthetic environments everywhere associated with this megafauna organisms, with this Rimicaris or the Riftia. And of course you can find them in nodule habitats. You can find them on the nodules. We have no evidence that some of them are living also in the nodules, but you find them on the nodules. You find them in the sediments, and you find them also parasitic or associated to the megafauna in the nodules areas. So what you need to know if you want to identify a copepod. Copepods are crustaceans. I forgot to mention that. So they're very small crustaceans in the range of 0.5 millimeter to 1 millimeter. But of course there are other size classes. So they have legs and limbs, and they have carapax, so they need to mold for growing. And we, taxonomies, we count the number of segments in the different appendages. So this is the first antennule. And you see there are males and females, so they have separate sexes. The males, they have an antenna that they can move and grasp. And the females, they have an antenna where they cannot use for grasping. So this is what we count when we identify the copepods. These are the mouth parts. They have mandibles. They have other mouth parts. They have kind of a brush. They have also some mouth parts that are also for grasping, like this maxillibed. They have also swimming legs. The swimming legs are characterized by having an endopodite and an exopodite, 2 rami, each of them with 3 segments in the adult stage. But these segments can also be modified, as you can see here, for grasping or attaching to the sediment or to bacterial mats or whatever. Very important is also how this seta, and you can see here the seta and spines, is the morphology of these elements. So you can have kind of a flexible seta or a spine, a laminate spine. So these are all the things that we are looking at when we want to identify the copepods. And also how many of them. This species has 4 spines, this species species has only 3 spines. These are all very, very important taxonomic characters. Also here you see males from the different copepod orders, and you see that there are some structures inside the males. This is what we call a spermatophore. So the sperms are housed into kind of a bottle, which is the spermatophore. And the whole evolution of the copepods have been, since the last 300 million years, has been influenced by how the male can grasp the female and transfer the spermatophore in order to reproduce. So, and they have invented a variety of modifications and strategies and everything in order to ensure reproduction. Here you see what happened. This is the abdomen of a female with a spermatophore attached to the genital field. When it is attached, then the spermia go into the female and are stored into a receptaculum seminis, and the female can then produce these eggs, egg sacs, with fertilized eggs. Different strategies have been developed. For instance, this pre-copulatory or post-copulatory mate guarding. What you see in this picture are 2 copepods. The male, you identify it with the grasping antennule, is grasping a female. And you see this female is smaller than the male. This is unusual because normally the females are bigger. But this female is a juvenile. So the males grasp a juvenile and goes with the juvenile until it molts to an adult in order to ensure that it is a virgin juvenile, a virgin female, and that he can be the first one putting the spermatophore and releasing the spermia and being the one reproducing or fecunding the eggs. There is also another strategy, which is the post-copulatory mate guarding, which you can see here in this picture. These are 2 adults. The male is grasping the fulca of the female. They have already copulated. The male have transferred the spermatophore. And what the male is then doing is waiting until the female produces the egg sacs. So he can be sure that these egg sacs were fecundated by him. So there's a couple of different strategies where to grasp the female for reproduction. After that, the copepods, they produce 6 nuclear stages, and then you have a metamorphosis, and you have another 6 copepodite stages. This is making things quite difficult because every copepod has, if you count all together, 16 different morphotypes. So they have all the different nuclear stages, they have the copepodite stages, but at copepodite 4 you can differentiate male and female, so you have 2 of them per sex. In total 16 morphotypes. If you take the number of copepods described for Europe, which is 3,600 species you can find in the European Register of Marine Species, this makes up 58,000 different morphotypes of copepods, which makes identification very, very difficult. Fortunately, the copepods, they follow German-Prussian rules in the development, and you have a very established pattern of segmentation of the limbs. In the first copepodite here, you see you have only the first and the second limb, with 2 rami and a bud for the third limbs. In the second copepodite, these 2 develop 2 segments, and a third limb is added, and a new bud is added, and so on and so on. So according to the number of segments and limbs, you can say exactly what kind of copepodite you have. In order to collect the copepod, at least for soft sediments, the multicora or the megacora is the gear of choice. Of course, this will not help if you want to collect copepods from hydrothermal vents. We have heard about this previously. Taking into account the relative abundance of copepods, this is a picture showing the relative abundance of Meiofauna organisms in the samples according to depth, and you will see that this is not changing very much. So we have the nematodes as the most abundant taxa, and the foraminifera if you count them, and then the relative abundance of copepods and naupliae are fair the same, like about 10%, something like that. So if you look at the number of individuals according to a depth gradient, you will see that you will find a decrease of copepods density in the sediments according to depth. But if you sample, for instance, as we did in the Benguela upwelling system, in the 5,000 meters in areas that have high productivity in the water column, so that the deep sediments, the deep sea sediments will get a lot of organic input. You see that you can reach densities of copepods which are similar to more shallow waters. So copepods density seems to be triggered really by the amount of food that they get from the water column, and this is really making the point. So we were also interested in knowing how copepods react to other changes in the environment. And I don't know if you know the Amperima event. So in the late 80s, there was an increase of the densities of these Amperima, these holothurians, in the porcupine abyssal plain. And the porcupine abyssal plain is actually one of the two sites that we know better because there are time series. So the UK colleagues are going there every year or every couple of years and following this time series, studying this abyssal plain. So we were interested to know how the copepods and Meiofauna in general react to this extremely increase in the abundance of the holothurians. As you can see here, the nematodes, this is the Amperima event here, the nematodes showed an increase after the Amperima event, but the copepods, they didn't really care. So there was no significant increase or reaction to the Amperima event across the years. Here you can see the years, 90 to 98, for the copepods. This contrasts also for what we found for the polypeds, where also you have an increase, and ostracods, even you had a decrease after the Amperima event. So if you want to know what is the seasonality of the abundance of copepods, also in the porcupine abyssal plain, you see these are the months of the years, and these are the values from the different cruises, and you see that there is no significant, there's a small trend, but there is no significant difference during the several different months across the year. You see a much better trend in the nematodes, so there are more seasonality in the nematodes abundance than the copepods. So the copepods don't really seem to care about seasonality or about other things that are going on, like the Amperima events. If we look, for instance, at the presence or absence of nodules, here we look in the French area, Nodinaut cruise, at the abundance of Meiofauna. The blue dots are area with nodules, not nodules, okay? And the green dots are areas without nodules. This is an MDS plot, and remember this position. So you have here at the bottom the areas without nodules, and at the top the area with nodules. If you see the abundance of nematodes, for instance, you see that the nematodes, they prefer the areas without nodules in terms of abundance. Yeah? But also the tardigrades. They like more the sediments without nodules, even though there are some in the area with nodules. But the copepods, they don't seem to care about that. Also the ostracods, they don't care about the presence of the nodules for the abundance. If we look up, however, in the community structure, we see that there are big differences in the families of copepods living in the nodule area compared to the non-nodule areas. Here that's a pizza diagram. You see that some families in this specific study, like these zosimids and the cantocampids, they are more common in the area without nodule than they are in the area with nodules, where they are virtually absent, at least the zosimids. So, how many species are there of copepods in the deep sea? Here you see a graph where we computed the number of individuals that we have in multicolor samples related to the number of species. And what you can see here is that this is a straight line. So this is a slope of 0.8. And the more individuals you look at, the more species you have. This is Atlantic data. So we were a little bit scared about that. And we made the same exercise with Pacific data and Indian Ocean data, and this is what we got. Exactly the same. You have also an increase of individuals, increase of species, linear relationship, a slope of 0.8. So the question is, where is this ending? So how many species are really there? In order to get more evidence for that, we said that, okay, we are going to look at two stations in the deep sea And we are going to take a lot of samples and look how many species we have. So we did this in the Angola Basin. We took at 2 stations 35 subsamples or multicolor samples, and the other station 40. We collected 7,000 hard-particulate copepods. 78% of them were copepodites, so these juveniles that we cannot really identify. We just work with the adults. So there were about 2,200 adults. Okay, much more males than females than males, and some of them were damaged. We could not identify them, so at the end we used 2,152 hard-particulate adults. And the scary movie about that is that this is the list of species. This is my colleague Georgi looking at the list of species. There were 682 species at only 2 stations, right? 2 stations in the Angola Basin. The northern station is more influenced by the Benguela upwelling system, so this is getting a lot of food input from the water column. This is the more oligotrophic site. 600 species, 172 species, only 90 species, 30% were in common between both stations. The most scaring thing about that is that 99.3% of the species were new to science, not described by anybody else. So, but if we look at the diversity, we see that the upper station, which has more food, has less diversity. This is the number of families than the most oligotrophic stations. They have many more different families. You see this also in the Jacquard index, the evilness index. The southern station with more oligotrophic has a higher evilness than the station with more food, even though the evilness is very, very high. It's 9.5. Many species, many people ask us, but these 600 species, I mean, how different are they? Maybe they are the same species, but you didn't notice that? No, they're really different. I'm just showing you here some pictures of some animals. You can see that the morphologies are very, very different. These are even different families, different genera. It's quite easy to differentiate them. Even if we have species from the same genera, like in this case, this Serviniella, there are always good characters, like look at this Fulca, short, this is twice as long, or the robustness of the legs. And if you start counting the segments and the seta and so on, you will see there are many, many differences between them. Of course, all these species, I need to say that probably they will never be described. This is a graph showing the time lag between discovery of species and description of species in the abyss that we computed from the SEDAMAR database. And most species are described typically between 6 and 8 years after collection. But we have a long tail here of species that they need even hundreds of years to be described. We computed this analyzing this database that we produced during the Census of Marine Life times. This is a database collecting 11,000 records for 3,000 species in the abyss. And also what we can learn from this database is that 50% of the species from the abyss have been described before 1920, and the other 50% have been described after 1960. So this is a cumulative curve. But if you see the descriptions per year, you see that after 1990, we have a drop of descriptions. And this is probably due to the loss of taxonomies, and probably will never reach the rate of description that we had in the past. So how to move forward? Maybe genetic methods are the tool. We are using genetic methods to identify copepods and to compare between sites. But at the end, you have the same picture. Here you see we identified 132 species of painted copepods with the 12S gene from the CCZ, the German area. And we found only 11 species that were found in more than one place. Most of the species that we have found, they are singletons, they are rare, and this database is increasing. Annika will talk about that. The next question is, are there different copepods in shallow water, hot vents, and abyssal plains, and so on? We have compiled here from literature the information about, at genus level, about 800 samples, 20 samples from hot vents, 9 samples from cold seeps, 84 samples from abyssal plain in the Atlantic, 39 samples from nodules areas, and 729 samples of shallow waters. And we end up with 184 genera. If you see at this ordination plot, this MDS based on the Euclidean distance on presence-absence data, you see that we have a good separation between the abyssal samples and the shallow water samples. The hot vents and the cold seeps, they cluster kind of in between abyssal and shallow water. And we also observe a separation between nodule areas and normal abyssal plains without nodules. So we calculated a model in order, the question is, can we use the genera that we find in the samples to know if the sample is the abyssal plain sample, shallow water sample, and so on? We calculate this random forest model, and yes, we can. Obama. So the error rate of this model is about 2.5%. Here you see the confusion matrix. This means that from 31 samples that were from abyssal nodules were classified indeed as abyssal nodules, but here you see that there is an error, a misclassification. The interesting thing here is that the misclassified sample is an abyssal. It was classified as abyssal, but it was a nodule. If you see the abyssal samples, most of them were classified correctly as abyssal, and only one was classified as nodule. But if you go to the cold seeps, very bad classification. Some of them were classified as cold seeps, some as hot vents, some as shallow, and so on. So you see that this fauna is in between. I will go back to this later. Here you see, okay, it's very small. You see which genera are important to classify the sample as a nodule sample, and you see the genera that are important for that, like Sarsameria, Paragesset, and so on. But one thing what is not so pretty for us is that you find genera like this Longipedia, which are shallow water genera. So why this genus is important to classify the sample as a nodule sample? Okay, you need to know that this modeling technique is a decision tree, and this decision tree may incorporate a genus that is absent from the target habitat, but if it is there, then the decision will say, okay, it's not that target area. So we studied also the copepods from caves. You give me an indication when I'm talking too much? Okay, thank you. Because I can't talk forever about copepods, so that's a problem sometimes, you know. Yeah. So we studied also the copepods from a marine cave in Marseille. You know, this is the 3PP cave. Marine caves, they are interesting because they harbor deep sea similar species or fauna. And here we had also the question from the reviewers, because we said that the fauna is similar to the deep sea, and the reviewers asked, how do you know that? Okay, in order to answer that to the reviewers, we trained another type of model, which is better for this kind of question, which is a naive Bayes classifier. Basically, you compute the probability using that database, that samples that I have shown here, that one. So you compute the probability that you find the genus Ameira present in an abyssal sample or in shallow sample, and absent in abyssal sample and in shallow samples. So this is so-called the conditional probability, and if you plot this in a graph, you can see that some genera, they have a high probability of being present in shallow sample, but low probability of being present in abyssal samples. And some of them, they have a high probability of being present in the abyssal samples, by low probability in the shallow samples. So once you have this, you can compute the posterior probability for every station along the transect in the cave, and we could reach to the conclusion, we can prove that the inner part of the cave have abyssal fauna, and the entrance of the cave has more shallow water fauna, and in between you have a mixed fauna. So what about the distribution of copepods in the abyss? These colleagues computed, observed 29 stations here from the Antarctic and the abyss of the South Atlantic, from the Diva and Andip cruises, and they looked at 311 paramesocrits. And they showed that some species, like this Glioxylus antipi, is present in the Antarctic, but also in the different basins in the Atlantic. The same applies for other genera. So they are widely distributed. In the next step, they looked at some species also in CCZ samples here, like this Emertonia, I cannot read it. And they found that some of the species are distributed across the Atlantic, but also they are present in the Pacific. But probably the most comprehensive analysis of the biogeography of deep sea copepods is done by Lena Menzel. This was a former student of us. Is now come back to our institute. She looked at these Mesocletodes copepods, which are deep sea copepods. And she looked at 770 samples from different oceans. So this was about 18,000 copepods. And for this family from the Algestides, she collected them from these 18,000, some 4,000 adult and copepodid species. It's just to show you, there were 2/3 of females, 1/3 of males. And of course, what she found is that many species were found, even though she looked at 18,000 specimens, many species were found just by singletons in one place, most commonly only one individual. But there were also some species that were widely distributed across the Atlantic, or even Atlantic and Pacific. So to show you only two examples, this is Mesocletodes abysicula, and this seems to be widely distributed across the Atlantic, Arctic Ocean, Mediterranean, or this Mesocletodes elmari, which is described from the CCZ, from the French claim, is also distributed here in the whole Atlantic. So there is some indication from some cosmopolitanism in deep sea copepods. In summary, she published a paper stating that submarine ridges, like the Walvis Ridge or the Guinea Ridge or whatever, they do not prevent the large-scale dispersal of abyssal fauna, in this case Mesocletodes. Here you see Lena with the model of his favorite Mesocletodes species. So now I move very quickly to the hydrothermal vents. Here the story is also more easy than in the, similar to the nematodes, we have less number of species, but there are almost 80 species described from hydrothermal vents. The dominant family is this Diribultidae. This is a siphonostomatoid copepod associated with the Meiofauna. So they live in the mussels, in the riftia, in the rimicaries. And here to give you an example, this is Stigopontius pectinatus. It has some modified seta on the antenna and the maxilliped. And this species is living in the gill chamber of rimicaries, which is this shrimp. We have different genera in different oceans. The highest diversity is here in the East Pacific rise, but this is also the place where we have more samples. But nevertheless, it seems that the highest diversity of genera is in the East Pacific rise. So we made a molecular study to confirm or to look at the molecular identification of the species compared to morphological identification. And we reached the conclusion that we could really match all. We had very nice clusters of molecular CO1 entities to known species. So we confirmed that the morphological identification was correct with the genera. And some of the species, they are restricted to some habitats. But we could also, looking at the haplotype diversity, see that there is a lot of dispersal along the ridges of these copepods, looking for one vent and moving to the other vent. If you look at the harpacticoid copepods in vents, this is also more easy. We have basically this genera, Patilaophonte, Xylora, San Mesocra, You find them in every bend. And this is, at first view, nothing spectacular because here you see a picture, Batilaofonte azorica. This is how they look like. The closest relative of Batilaofonte azorica is Batilaofonte peroensis, which was described 1902 from decaying wood perforated by boring mollusks, Xylophaga dorsalis, in the Feroe Channel, shallow water. So the hydrothermal vent harpacticoids, they are associated, they are related to fauna which you can find in shallow waters living on wood, decaying wood. The same applies for Xylora batialis. Yeah, this is the most common in hydrothermal vents. Xylora batialis was described by Hicks, 1988, together with Xylora neritica, from waterlogged decaying wood. This also from decaying wood in deep waters or in shallow waters. And that's it. Thank you very much for your attention.
I think we have time for some questions before lunch. I have to say, I didn't expect that we would have to put an X rating on some of your slides. I think we learn more about Copepods' sex life than we possibly wanted to know, really.
I also have some videos.
Oh, really? I suggest you approach him after the show. Questions? Simon?
That was very, very informative. Thank you very much. Given that we assume that copepods are less diverse than nematodes, and your copepod species accumulation curves go like that, what do you think the nematode species accumulation curves will do?
I don't know. There is the general impression that nematodes are more diverse than copepods, but this is something that we still need to check. Of course, in a sample, in one sample of multicolor sample, whatever, you have many more nematodes than copepods. So if you are looking at species density, how many species you find per unit area, probably you will have more nematodes.
Yeah.
If you are looking at species richness, if you relate the number of species to the number of individuals, I don't know. We have really never done this exercise.
But I think that exercise was never done indeed for nematodes. I agree with you. In a sample, you will get, in a sample of 10 square centimeter or 30 square centimeter, you will get more nematode species. But what we don't really know is the turnover, because there are so many species in one sample that we never arrive at describing them properly. So we end up with this species list A, B, C, D, and we can't compare the A, B, C, D from one area with ones of other areas.
You know,
So we don't really know.
yeah, historically, the methods of studying the nematodes and the copepods differ in the sense that because we don't have many copepods in the sample, we have 50, 60, 100, whatever. We look at all the copepods. And because there are so many nematodes, historically they just look at 100 or 200 individuals, and they calculate this estimated number of species by 51 individuals. You know, they don't look at the whole sample. So that's the difference.
Well, first of all, I really admire your work. You know, so for the copepods, each species they have so many morphotypes. Yet you people, you know, that manage to key to the species. So one of my questions, you know, in the past I asked Hans Dams, you know, so that how the environmental condition would shape the morphology of the particular species. I'm talking about plasticity.
Plasticity.
Yeah.
They don't have really much.
They don't change at all.
No, because
under
they
different
have
kind of environment.
They have. No, okay. You're talking about different species.
The same
Or
species.
Same species plasticity. No, they don't change. They don't change because they need to mold, and they have only 12 molds. This is also strict, and after that they don't mold anymore. So there is very little variation within the species. You may have one zeta less or whatever, but not really plasticity as we know from. So they are strict to the molds and what happened there.
Pedro, of course, everybody appreciates your work and there's no doubt about it. I personally feel jealous about it, how much work you have done it in the time.
Okay.
So thank you very much for that.
Actually, I didn't do almost anything. It was my people, my student, my working lab. So all the merits are.
What you don't see is the huge army of people standing
Yeah,
there.
yeah.
He is a
Behind me there are
he is a
many
direct- he has been a director of the institute. In spite of that, this was the data he could produce it. That's what one should appreciate it. That's what I wanted to say. One thing with related to this workshop, you mentioned that forget about nematode. Copepods don't care about the nodule. What would you like to just say something more about that?
Yes. Well, the number of copepods that we find, these don't seem to be influenced by the abundance of nodules. But Annika will talk more about this in her presentation because she has much more data to show that. But of course, there are different. There are other dominance patterns. So there are other families or genera more common in the nodule areas than in the areas without nodules.
Of course, it doesn't have direct this thing. Presently, you can't say. Can we say something? Mining will not have effect on
I think
Too early to say, but
yeah. No, I think it will because well, there are many aspects how you will change the environment with mining, and you will not only remove the nodules, but they will be also, you will change the structure of the sediments, you will compact the sediments, and so on. So there will be a couple of other impacts that will also affect the copepods, the community.
First of all, congratulations. I also like those slides for producing specimens. They are interesting. I wonder, your distances, it seems that these animals, the copepods, can just disperse widely. And you were mentioning driftwood as one possibility. But another would be, I don't know, larvae?
Yes.
Larvae distribution in
Yes.
the water columns, that would be a possibility.
Yes. Of course, yeah. Okay. They also emerge. They come out of the sediments, which nematodes normally don't like to do. But the copepods, they like to come out of the sediment once in a while, and probably they are then transported by the bottom currents. They drift, yeah.
Oh, Pedro, you come up with this old story about the deep sea copepod related to some copepod in the caves. Have you any news about that? We hear always that we tell our students that we can find this marine deep sea animals, not only copepod, in the cave. Why? Have you a new explanation? Because this weather is dark in there and cold in there, and so they're not really fit. Have you any idea why? To clearly show how deeper you come into the cave, the more abyssal
Yes.
copepod.
This specific cave is open.
Yes.
Right? There are ankyline caves where they are closed, so this is probably a different story. But this specific cave is open. This is in the Gulf of Marseille, and there are some upwellings once in a while every year where the water. You see that because nobody's in the water. They are all lying in the sun, but nobody's going into the water because the water is very cold in these episodes. So probably with this upwelling of deep water from the Mediterranean, some larvae or whatever also managed to get into the cave, and they managed to survive and produce some populations in the inner parts of the cave. This is the explanation that we have at the moment for that cave. You know that there is also a coronal tardigrade described.
Oh yes, of course I know that.
Which they only live in the abyssal plains, but there is only one species living in the cave.
Exactly, same cave.
Yeah.
Yeah.
And,
We don't have them in Yucatan.
You know, you have them.
No, we don't have deep sea animals in the caves. They are all shallow from the beach.
and the abyssal and the animals that we found inside the cave, this 3PP cave, they are really abyssal genera and families and so on. Sorry?
I'm not suggesting that you know the answer to this question, but I think somebody in the room probably will do. People keep talking about the biodiversity of Meiofauna associated with the nodule areas. What geological or ecological features actually give rise to those areas? Because I guess from a mining perspective, we're interested in a pristine nodule area versus a damaged nodule area. But why are the communities different, right? Where the nodules I don't even know what they do. Pop up, grow, emerge, exude, seep.
No. But there are specific bacterial communities associated to the nodules. So there are these manganese-reducing bacteria. So the bacterial communities, they are completely different in the nodules and around the sediments of the nodule than the bacterial communities from the sediment. So they are more similar to the bacterial communities in the crust on some seamounts. This may be, I mean, some nematodes or whatever organisms are feeding on that bacteria. It may be that this make a big difference. Also the 3D structure. So you have seen that copepods, they have some modified legs to grasp, to attach to sediments or filaments. And of course, these animals don't live or don't like to live inside the sediment. They want to live on the structures and move and grasp and so on. So if you don't have these structures, probably they will not be there. In the crevices of the nodules, they move there.
Any other questions? In that case, I think we can break for lunch. Lunch is through